Awesome, you guys. Okay, so let's go ahead with chapter 11. Here we're talking about physical and chemical control of microbes. How do we kill microbes and keep them at bay? There are historical ways that we have treated mainly food products, to control microorganisms. We salt foods, smoke foods, pickle foods, dry foods. So our foods are preserved, right? These are ancient ways of preserving food before refrigeration was, invented. Also, you could expose things to sunlight. We all know that UV damages bacterium, and they also dry out. So the combination of drying out and UV kills the microbes. Burning clothing, burning corpses. The people burn their dead. This helps to prevent the spread of disease. Storing water in copper and silver containers. Did you know that heavy metals are toxic to microorganisms? So by keeping your water in these metal containers, you actually keep it relatively germ-free. By the pro tip for those of you that have pets too, if you have a metal bowl, that's better than a plastic bowl for your pets too, for their water, because again, those heavy metals are antimicrobial in nature. Now, these are terms we should know about microbial control. These terms may seem similar, but they mean distinct different things. So for instance, sterilization. When I say sterilization, that is a whole different beast than saying disinfection, right? Those are two completely different terms, but mostly people use them interchangeably, right? People don't know the difference between disinfect and sterilize. But if you're going into healthcare, you should definitely know the difference between disinfect and sterilize. Sterilize is the gold standard, okay? It's the gold standard. Sterilize means you've killed anything on that object that could possibly cause disease, right? So any organism, any endospore, any fungal spore, any bacteria, protozoa, whatever. Sterilization is, difficult to achieve. You need something like literally fire. This is why we flame our loop, right? That sterilizes the loop. So after you flame the loop, that's a sterile loop. An autoclave, we're going to talk about what an autoclave is, but an autoclave is another way to sterilize something. But that's the gold standard. Under sterilized, the next level down is disinfect, right? Disinfect is a less stringent requirement. When you disinfect something, you could kill, 99.99% of all possible pathogens, but you're never sure. It's not 100%, right? And usually it's the endospores that you missed, right? You ever seen a Lysol bottle? Does it say kills 100% of everything on the Lysol bottle, or does it say 99.99%, right? So Lysol is an example of a disinfectant, right? It might kill 99.99% of germs, but guess what? It probably didn't kill any endospores or many endospores, right? Does that make sense? Antisepsis, also known as degermation. Antisepsis is when you add some kind of disinfectant, but on a living tissue. So have you ever heard of antiseptic mouthwash? Mm-hmm. Right? Why do you think they call it antiseptic mouthwash and not disinfectant mouthwash? Well, because it's going on your living tissue. It's going in your mouth. Your mouth is a living tissue, right? Inside of there, living tissues. You got your tongue, you got your cheeks. If you're applying a chemical agent to a tissue, a living tissue, it's an antiseptic, okay? Sometimes you could use something as a disinfectant or an antiseptic. So for example, people use hydrogen peroxide. You're not supposed to because it causes a lot of damage, right? They say don't use hydrogen peroxide, but they used to use hydrogen peroxide on the skin, right? And so if you're using hydrogen peroxide on your skin, you're using it as a antiseptic, right? But if you're using hydrogen peroxide on an object, then you're using it as a disinfectant. Does that make sense? You see the difference there? And then the lowest stringency, the lowest requirement is decontamination, also known as sanitation. This is where you can use mechanical removal of most microbes. You wipe something off, or you wash dishes with some regular soap. It's not very stringent, right? It's just you got most of those microbes off of there, right? So when you do your dishes, you're sanitizing, you're decontaminating. You're not, that's the lowest health standard. Okay. And again, why do we need microbial control? Well, because we learned early this semester that microbes are ubiquitous. Bacteria is ubiquitous. That means everywhere. There are fungal spores everywhere. Bacterial spores are pretty abundant. There's protozoa. I mean, there's microbes. These microbes might make you sick, right? Especially in the healthcare setting where certain instruments might be used to probe you or to cut you open, right? So we need to understand how to sterilize those types of, instruments, right? And here you can see different organisms have different resistances, okay? Not all microorganisms are as easily killed as, let's say, enveloped viruses. You see, enveloped viruses are the easiest to destroy. They're the least resistant to antimicrobial techniques, right, and methods. But on the other end of this scale, prions, look at them. They are really hard to treat, really hard to destroy. I just saw a story about a lab that was working with prions. Did you guys hear about this story? There's a lab that was working with prions, and 2 of the former lab members came down with basically Creutzfeldt-Jakob disease. They came down with the prion disorder years later. So, and it's still a mystery, like how they caught it from the lab, because they didn't eat anything. One of them may have caught it through a break in the glove, like they broke the glove and it cut right through the blood. The other one, they still don't know how he caught it. So, we still don't fully understand prions, and they're the hardest ones to treat because it's just a denatured protein. What are you going to do? Denature a denatured protein even more? It's really hard to treat, right? And also endospores, right? Endospores are really hard to treat. You see that? Endospores, very difficult. This is why we need fire. This is why we need, steam. We need that autoclave. And again, it's not sterile unless you've destroyed all of these, right? Every single one. Then it's sterile. Okay. Endospores are a huge problem. Remember, destruction of endospores is the goal of sterilization. Okay. Again, does boiling water sterilize? Do you guys know? No,
Yes.
no, not at all. No. No. The boiling water destroys many organisms, but it's more disinfecting. It's not sterilizing. Endospores laugh in the face of boiling. So, you can boil all you want. The endospores are still viable. And so you cannot sterilize with boiling. Here we can see different types of methods, different methods of disinfecting or sterilizing, and how long it takes to kill the vegetative cells and the endospores. Right. So here you can see with moist heat, this is an autoclave. When they say moist heat, they mean steam. And this is an autoclave. An autoclave reaches temperatures of 120 degrees C. Okay. Vegetative cells die at around 80 degrees C. Endospores, they are at least 1.5 times more resistant than the vegetative cells. So it takes a lot longer to kill endospores than it does vegetative cells. Okay. Same thing with X-rays. X-rays, it takes a lot more grays of X-ray radiation to kill the endospores than it does vegetative cells. It takes a lot more sterilizing gas to kill endospores than regular vegetative cells. It takes a lot more glutaraldehyde, which is a sterilant cross-linking agent. It takes it 3 hours to kill endospores as compared to 10 minutes with vegetative cells. So if you notice, all of these, it takes a lot more firepower, right, to kill the endospores than it does to kill the vegetative cells, right? Okay. By the way, did you guys know when you boil water, what temperature does it boil at? Water, boiling water. 100 degrees C, right? 100 degrees C. But now if you crank it up, so the water's boiling, and then you crank up your stove even more. So now it's boiling even more. In fact, it's on maximum. What's the temperature of the water? It's still 100 degrees C. You can't boil water higher than 100 degrees C. It doesn't matter if it's boiling only a little. It doesn't matter if it's boiling like crazy. It's still 100 degrees C. You didn't go above it. Okay? The only way to increase the temperature of water past 100 degrees C is to do what? Pressure. Like a pressure cooker, you know? If you prevent the steam, steam from escaping, and you pressurize it so the steam can't escape, then the temperature of that steam can reach 120, right? That's what a autoclave is. An autoclave is basically a steamer. It's a pressure cooker, you know? You ever cook with a pressure cooker? It fixes your food in no time. Like, your food's ready in no time. Because in a pressure cooker, the temperature went above 100 degrees, so you cooked your food faster, right? An autoclave is like a glorified pressure cooker. An autoclave is basically this pressure cooker that gets the moist heat up to 120 and increases the pressure, and the combination of the pressure and the steam kills all endospores, everything, right? So it sterilizes. Again, sterilization is a process that destroys or removes all viable microorganisms, including viruses, including endospores, everything. Any material that has been subjected to this process is said to be sterile. So when you pass someone a sterile scalpel, that means it's been autoclaved, right? Now, if I leave that sterile scalpel out on a table for a while, is it sterile anymore? No. The second even one particle of dust lands on that scalpel is no longer sterile. Isn't that interesting? So the second you touch that scalpel accidentally to the table, or you touch here, it's not sterile, right? It's only sterile if it's been autoclaved and it's inside that autoclaved packaging, right? When you bring it out and leave it out, no longer sterile. If you accidentally touch something for a second, no longer sterile, right? So what products need to be sterilized? Does your food need to be sterilized? No, that's not necessary, right? But there are certain things that we need to sterilize, right? So surgical equipments, right? So if we're going to do a surgery on you, all of those tools need to be sterilized, sterile. Syringes, right? Syringes need to be sterile. You take the needle out of its sterile packaging, you need to use it and then dispose of it immediately, right? And then some packaged foods, but these are usually for, MREs or some kind of long-term shelf life stuff. And for canned goods too. You don't want your canned goods to have endospores, because then you could get botulism. Remember that? What about disinfection? Remember, disinfection is the next level down from sterilized. It's not as stringent, but it's sufficient for most cases, right? In your kitchen, do you need to sterilize your countertops? Not necessarily. You can disinfect your countertops. Just spray them down with Lysol. That's plenty sufficient for something like that, okay? So you're killing vegetative pathogens, but not necessarily many endospores when you do this type of disinfection. Okay. Disinfectants are used on inanimate objects, right? Like a table. Whereas antiseptics, remember, antiseptics are used on living tissue. Antiseptics are used on living tissue. Okay. We learned about aseptic technique, right? Aseptic technique, the sterile method of transferring microbes or dealing with microbes in a way that prevents contamination and prevents breaking containment. Some of these chemicals we work with, these antimicrobial chemicals, are called cidal agents, like bactericidal or sporocidal. You know, what does that suffix mean? It means kills, right? Kills. So if something is bactericidal, it kills bacteria, right? If something is sporocidal, that means it kills spores. Fungicides kill fungi, right? Anytime you see cide, that means kills. Now, virucide is kind of a misnomer because viruses aren't alive, so you can't kill them. However, they're still called virucides, but just understand you're not killing the virus, you're inactivating the virus, right? Because there's the active viroid, the active virus, then the inactive virus, the denatured virus. So virucides, they don't kill the virus, they inactivate the virus. And a germicide or a microbiocide, these are just general terms for, you know, like Lysol would be a germicide. It just kills a bunch of different microorganisms. And by the way, are fungal spores as hard to kill as bacterial spores? No, right? Fungal spores aren't super tough. They're not super tough like endospores are. Fungal spores are designed to disseminate easily and spread and just for reproduction, right? They're more reproduction oriented and less defense oriented. So it's way easier to kill a fungal spore than it is an endospore, a bacterial endospore. Those are the really tough little buggers to kill. So what are static agents? The suffix static. Static means not moving, right? So static agents prevent the organism from dividing. It prevents the organism from growing, right? So if I treat you with a fungistatic chemical, it's not necessarily going to kill the fungi. It will keep the fungi from growing, right? Or a microbiostatic agent will keep the microbe from growing. Or a bacteriostatic agent keeps bacteria from growing. Sometimes all you need is to keep the infection from growing and then allow your immune system to do the rest, right? Your immune system kills the infection, but at least you prevent it from spreading, right? That's sometimes sufficient. You don't always have to kill the microorganism. Many agents are static. And what about decontamination or sanitation? Remember, this is the lowest standard. This is the lowest stringency. Okay? This is what they do in general health practice, like at restaurants and stuff. They're required to wash the dishes or scrape the food off and then rinse it off. This is just the lowest standard, okay, to meet health requirements. they say it reduces contamination to a safe level, right? To a safe level. Okay, and so when you go on to, when you go on to healthcare, you're going to have to ask yourself, does this instrument need to be sterilized, or can I get away with it being disinfected? How do I treat this, or can I just sanitize it? And it all depends. Like, is it gonna, be in proximity to the patient, or is it going to be used inside of the patient's organs? You have to assess the level of control you need for each, scenario, right? So does the item need sterilization? If I'm giving someone a spoon so they can eat their Jell-O, does it need sterilization? No, obviously not. It just needs sanitation, right? But if I'm handing the doctor a scalpel so they can do an operation, then it needs to be sterilized. So, this slide is basically asking you what, there's no cookie cutter way to go about things. You have to assess each scenario differently. Here I need to sterilize, here I don't, here I need to disinfect, here I can sanitize. Again, it all has to do with how likely is it to cause disease in the patient, right? So look, a critical medical device is expected to come into contact with sterile tissues. Remember, inside of your body, inside of your tissues, inside of your fluids, right? So let's say you're putting a pacemaker in a patient, right? You want to make sure that pacemaker is sterile, right? Yeah, for sure, right? So that needs to be sterile, right? You can't just put, this random pacemaker into a person without ensuring that it's sterile. But what about a semi-critical medical device? It could come into contact with mucosal membranes. So, you have a feeding tube, or something like that. That would be a semi-critical medical device. It's not inside of your tissues, but it's among your membranes. It goes through your nose and everything. What about non-critical medical devices? Those that do not touch the patient and are only expected to touch the skin. So, like those little pads for monitoring your EKG or whatever, that would be a non-critical. So you have to ask yourself, to what extent is this device going to, interact with my patient? Obviously it needs to be sterile if it's a critical medical device, right? And then the goal of all of this is to kill microorganisms, right? The goal of control is to kill the microorganisms. But it's hard to detect whether you've killed the microorganisms because you can't even see them, right? I don't know if there's living bacteria on this table or dead bacteria on this table. I don't know. I can't see it. I'd have to do some studies. So it's hard to see if, organisms are dead or alive. Okay. Why? Because even if I spray it with Lysol, even if I spray the table with Lysol, I still can't tell if anything's dead or alive because it doesn't alter the overt appearance of the microbial cells. Even if I did take those cells and put them under the microscope, even if they were dead, they would still look like cells if I stained them. So it's a pain because you just have to rely on your techniques, right? You have to rely on doing things correctly. So for instance, you guys know I say spray down your table and wait how long? 5 minutes, right? Why do I say that? Well, because they've done studies to show that if you leave that disinfectant on for at least 5 minutes, then you've achieved good disinfection. Then you've killed 99.99% of everything, right? But if you just spray the table and wipe it immediately, they've shown that you may kill far less than 99.99%, right? But that's why you just have to go off of what it says. So when you go into the clinic and they say, did you know a common sterilant in the clinic is this sporicidal liquid glutaraldehyde, right? Glutaraldehyde. they might say, you know what, this patient in room 5, this patient had C. diff. Remember the endospore forming C. diff. And we're concerned that it's like contaminated the room, right? So why don't you spray it down with glutaraldehyde and wait 3 hours, right? Wait 3 hours. Would it make sense to just be like, whatever, I'll just wait 10 minutes? You know, yeah, if you wait 10 minutes, you've killed the vegetative cells, but you've really not killed the problem. Then, you know, you wipe it up, you're like, whatever, it should work, you know. The next patient comes in, those spores are still intact, they touch something, they touch their food, they touch their mouth. Next thing you know, the next patient has C. diff, right? Because you didn't wait the full 3 hours. So again, you can't see on a table if these spores are living, dead, what, you know? So you have to go off of what it says on the label of the disinfectant you're using. There's a reason why Lysol says, you know, wait a minute and then wipe it. There's a reason why, you know, in your lab, the disinfectant says wait 5 minutes and then wipe it. There's a reason why glutaraldehyde needs to be left on for 3 hours and then wipe it, because they've done all these lab experiments. They've done all these tests, and that's what their data showed, right? So try to adhere to those things.
Now let's talk about factors that affect the death rate of microorganisms. The number of microorganisms is important. If you're trying to destroy billions or trillions of microorganisms, obviously that's going to be harder than a few dozen. The nature of the microbes in the population, are they endospore formers? Are they easily disrupted? Are they easy to kill, or are they more hardy? Do they have, let's say, mycolic acids? Are they acid fast? Those are typically harder to kill off. The type of microbial growth. Are these microbes in a biofilm, for instance? That's going to be tougher to kill off. Temperature and pH of the environment can play a role. The concentration of the agent. So the chemical you're using, are you using a strong concentration, a high concentration of, let's say, bleach? Or are you using diluted bleach? That could play a role in the death rate. The mode of action of the agent. What part of the cell does that agent attack? That could take account as well. The presence of solvents, interfering organic matter, and inhibitors. All of these things can affect the death rate, so you have to take them into account. And by the way, speaking of the death rate, these microorganisms, when they die off in a logarithmic fashion. This means that when you add a disinfectant, when you add an antimicrobial chemical, not all the microbes die instantaneously. They die off in a logarithmic fashion. They die off over time in a logarithmic fashion. So this is something to be aware of. They don't instantaneously die. Instead, they die off logarithmically. Now, how do antimicrobial agents work? Typically, these agents target either the cell wall or the cell's cytoplasmic membrane, or either DNA or RNA synthesis by targeting RNA polymerase or DNA polymerase, or they can even target protein synthesis. For instance, they could target the ribosomes, which synthesize the proteins. These are typical targets of antimicrobial agents. And this can lead to all kinds of effects. If we're talking about agents on the cell wall, well, if you block cell wall synthesis, you could have a negative or deleterious effect. You could cause the cell to perhaps lyse. This is how penicillin works. Penicillin prevents the maintenance of the cell wall of bacterium, and this results in osmotic stress. Water forces its way through osmosis into the cell, causing the cell to lyse. Some of these agents can even digest the cell wall of peptidoglycan, break down the surface of the cell wall. Any of these could be fatal for the cell. And how about the cell membrane? I told you about the membrane of the cell. We talked about surfactants, which can insert themselves into the membrane of the cell, and this can cause the cell to lyse or break open. These cause the cell to become more permeable. They can lose their selective permeability. This means that vital innards could flow out of the cell, and the cell lyses. This is a damaged cell. And remember, again, surfactants are an example of chemicals that disrupt the cell membrane, opening up leaky spots that really damage the cell, allow damaging chemicals to seep into the cell, and important ions and important cellular components to leak out. Here you can see how the surfactants work. Again, the surfactants enter into the cell membrane, disrupting the cell membrane, opening up these leaky spots. And what about agents that affect protein or nucleic acid synthesis? These are also typical targets of antimicrobial chemicals. Substances that inhibit ribosomes, they're going to inhibit protein synthesis, and then the cell can't divide. These are typically static agents. Static agents prevent the cell from dividing. Also, nucleic acid blockers. These are agents that can impede transcription of DNA into mRNA or even DNA replication. These typically act on DNA polymerase, the enzyme that copies DNA, or RNA polymerase, the enzyme that transcribes DNA into mRNA. Either way, you're not going to allow the cell to grow. And remember, some of these microbial agents, they can attack the proteins themselves. Remember that a protein needs to be folded in order to function. And so when a cell produces proteins, that protein needs to fold into a native state, and that protein then functions normally. But several of these chemicals, including, like, let's say alcohol, for instance, can denature proteins, causing the protein to misfold and become non-functional, and that can lead to cellular death. Again, agents that alter protein function, typically they cause proteins to misfold. They cause these proteins to denature. Once a protein is denatured, that means the protein can no longer function and the cell usually dies. Moist heat can cause this denaturation of proteins. Also, strong organic solvents such as alcohols or acids or phenolics, these can have the same effect. Here you can see the native state or the properly folded protein, which would be functional, and then the denatured protein, the protein that has become unfolded. Once a protein becomes denatured or unfolded, it can no longer function, and it can no longer renature and pick up function again. Next, let's discuss methods of physical control. Moving on from chemical control to physical control. This would include methods like radiation, filtration, ultrasonic waves, as well as cold. Now, starting with heat as an agent, specifically moist heat. Moist heat can refer to water, it can refer to steam. Hot water, boiling water, and steam. The temperature ranges that would destroy a typical mesophile, or a type of bacteria that would usually cause human disease, is about between 60 degrees C to 135 degrees C with water or steam. The temperature of steam can be regulated by adjusting its pressure in a closed container. Water boils at 100 degrees C, and no matter how much you turn up the temperature, the water will not rise above 100 degrees C. It will simply become steam faster and faster. So if you want the temperature of water to increase, if you want moist heat to be warmer than 100 degrees C, what you need to do is apply pressure, like a pressure cooker. And essentially, that's what an autoclave is. An autoclave is a pressurized chamber that allows water to form steam, and that steam can reach 120 to 135 degrees C, and that's sufficient moist heat to sterilize. Now, the reason you sterilize is because that hot temperature, that really warm temperature, causes coagulation and denaturation of proteins. Now, what about dry heat? Dry heat is when either air with low moisture content or a flame or electric heating coil is applied to the sample. This is a dry heat. So when we flame our loops in the lab, or we use incinerators or flames to incinerate our loops, this is an example of dry heat. It dehydrates the cell, it oxidizes cellular components, and it burns them. Oh, there's Wicket behind me. I think you guys saw him walking behind me. Oh, that's Gizmo. Gizmo, come say hi. Gizmo's back there adventuring. So again, let's compare moist heat to dry heat. Moist heat, remember, to reach temperatures of 121 to 134 degrees with moist heat with water, you're talking about a pressurized container. You're talking about an autoclave. And this can sterilize within, just minutes. At 121 degrees in an autoclave, sterilization is achieved in 15 minutes. And this means that even endospores are destroyed in 15 minutes. And the warmer you go, the less you can, the less time is required for sterilization. With dry heat, however, at the same exact temperatures, dry heat takes longer. Okay, dry heat takes longer to sterilize, but it is possible to sterilize with the same heat. The main point of sterilization is to kill even endospores. Endospores have the greatest resistance to disinfection and to heating because of their, dried out seed-like nature. They are very tough structures. And the point of sterilization is to destroy endospores. Vegetative cells are far easier to destroy and to kill off. Vegetative cells can vary in their sensitivity to heat, but it's typically nowhere near as robust as an endospore. Now, both temperature and length of exposure should be considered for adequate sterilization. We need to have the right temperature for the right amount of time to kill off the microbes. At higher temperatures, this would allow shorter exposure times. Lower temperatures, conversely, require longer exposure times. Each organism has two distinct times, the thermal death time and the thermal death point. I want to explain the difference here. The thermal death time of an organism is the shortest length of time required to kill all test microbes at a specified temperature, while the thermal death point is the lowest temperature required to kill all microbes in a sample in 10 minutes. So let me explain the difference. For instance, let's say we're trying to kill off E. coli, and I put E. coli at, 55 degrees C. And then we notice that it takes an hour to kill all of the E. coli. That would mean that at 55 degrees C, the thermal death time for E. coli is 1 hour. Does that make sense? But we have not reached the thermal death point. The thermal death point is the temperature where all of the E. coli would die in 10 minutes. So let's say we did the experiment again at 100 degrees C, and then we saw that all of the E. coli were dead before 10 minutes, we could call that the thermal death point. Does that make sense? So the thermal death time is how long does it take for all of the microbes to die at a particular temperature, and the thermal death point is the lowest temperature that kills all of them in 10 minutes. And remember, we said that an example of moist heat is boiling water. I want you to know that boiling water is good for disinfection, but not for sterilization. Boiling water is not a sufficient means for sterilization. This is the problem. 100 degrees C will not kill all resistant cells, nor will it kill most endospores. So why do we boil water? It is useful in the home for disinfecting water, killing most pathogens, but remember, you are not sterilizing anything by boiling it. You are simply reducing the bacterial load, but not sterilizing. Pasteurization is an even lower standard. With pasteurization, we want to heat a particular food or beverage to retain its flavor. This means that we cannot heat the food past a particular temperature. We all know what happens when you boil milk, for instance, right? If you boil milk, then it tastes off. It does not taste right. So with pasteurization, which was developed by Louis Pasteur, here we go to, either 71.6 degrees C for 15 seconds in a flash method, or in a batch method to 63 to 66 degrees for 30 minutes. You see, this is well below boiling, but it's sufficient temperature for denaturing proteins of bacterium. So it can inactivate most viruses and destroy the vegetative stages of 97 to 99% of bacteria and fungi. However, it does not kill endospore, and it does not kill heat-resistant microbes. But remember, the main point here is not to sterilize the beverage or the food. Here, the main point is to retain the flavor and reduce the microbial loads. And so it's nice for milk. When we pasteurize milk, wine, beer, and other beverages, we reduce the microbial load and retain flavor. Now, if you do want to sterilize, we want to use steam under pressure, which can achieve sterilization. And we do this with a pressure cooker called an autoclave. With an autoclave, this is a device that can subject pure steam to pressures greater than 1 atmosphere. We're going to 15 psi of pressure for 20 minutes, and this achieves a temperature of around 121 degrees C. The steam is very warm, and this is sufficient to sterilize glassware, cloth, medical instruments, liquids, etc. Anything that's heat resistant, we can put in an autoclave for sterilization. And yes, this means that endospores are neutralized. Here is an image of the inner workings of a standing autoclave. Essentially, it's a pressure cooker. You place your samples in this chamber. There's steam at high pressure, reaching a temperature of 121 degrees C for 15 psi for 20 minutes, 15 to 20 minutes. That's sufficient to sterilize. And so in the clinic, in the dentist's office, even at tattoo artists, tattoo parlors, you will find autoclaves where instruments are sterilized. Now remember, for dry heat methods, incineration, here we're using, for instance, a flame. We can use a Bunsen burner. A Bunsen burner reaches 1,870 degrees C at its hottest point. And this is more than enough to incinerate, to destroy all living organisms or all viable cells, including endospores from your instruments, including your inoculation loop, your inoculation needle, etc. This is common in the lab. You can also use a hot air oven, a low moisture environment, an oven, to sterilize. But remember, with dry heat, it takes quite a bit of time, hours to sterilize. Now, what about cold? One thing you should know about cold is that cold does not kill the microorganisms. Instead, cold merely slows down the microbes. By placing microbes, let's say, in a freezer at minus 20 degrees C, you're not actually killing microorganisms. You're making them slow to the point of suspended animation. They stop moving. That's essentially all that's happening. So we're not using cold to kill microbes. Cold is lower. The freezer usually is lower than the minimum temperature required for growth of microbes. And so these microbes simply stop growing, but they're not dead. What about desiccation? What physical control method is this? What does this refer to? Desiccation refers to dehydration. A desiccant is a substance that removes moisture from the air. So desiccation is drying of the cells. Remember that all cells require water for life, and most of the volume of a cell is water. So by drying the cell, by drying the environment, these pathogens can't grow. Desiccation can also preserve foods. So if we make beef jerky, if we make dried fruits like dried bananas or dried apricots, these foods are resistant to microbial growth because they do not possess enough water for the microbes to grow on. Next, what about radiation? This is another form of physical control, radiation. Radiation refers to energy emitted from atomic activities and dispersed at high velocity through matter or space. There are 3 types of radiation that we can use for microbial control: gamma rays, X-rays, and ultraviolet radiation. Irradiation refers to cellular bombardment with radiation. So if I use radiation to bombard cells and kill cells, I'm irradiating those cells. Ionizing radiation, this type of radiation causes catastrophic mutations in DNA. It causes double-strand breaks in the DNA. And X-rays and gamma rays are capable of ionizing radiation. Conversely, non-ionizing radiation causes these thymine dimers to form, or cytosine dimers to form in the DNA. This is known as abnormal bonding mutations. And UV radiation is known for causing this type of non-ionizing damage. Now, ionizing radiation can sterilize without heating. This is also known as cold sterilization, but don't confuse cold sterilization with sterilizing with cold. The reason it's called cold sterilization is because gamma rays, X-rays, and UV rays, they don't require a heat, and so it's known as cold sterilization. So that's why it's called cold sterilization. So there are devices that emit ionizing rays: gamma ray machines, X-ray machines, cathode ray machines. These machines emit ionizing rays, which can cause double-strand breaks in the DNA, causing cellular death and sterilization. Again, ionizing radiation includes gamma rays and X-rays. This causes double-strand breaks to form because of the stripping of electrons from DNA and RNA. Non-ionizing radiation, this includes UV rays. This leads to abnormal bonds within the DNA, such as thymine dimers or cytosine dimers. Together, those are known as pyrimidine dimers. because thymine and cytosine are known as pyrimidines. UV rays are a form of non-ionizing radiation. These rays are absorbed by DNA and cause thymine-thymine dimers or cytosine-cytosine dimers. These are not as penetrating as ionizing radiation. So with UV, did you know with UV, the bacteria have to be directly struck by UV. They have to be on a smooth, clean surface, and the UV rays have to directly penetrate the cell. If the cells are underneath plastic or underwater, then UV will not have an effect on those cells. The UV has to directly touch the cell, and the cell will develop these thymine-thymine dimers or cytosine-cytosine dimers, and that will lead in cellular death. So here you can see exactly what I've been talking about with UV. Here you can see two adjacent thymines in the DNA molecule, thymine, thymine. And here you can see the effect of UV. The effect of UV is forming a dimer between these two residues. UV radiation can also disrupt cells by generating free radicals. Free radicals are these toxic photochemical products that damage the cell. They interfere with essential cell processes by binding to DNA, RNA, and proteins. This can lead to cell death. And lastly, let's talk about filtration. We can use the physical control method of filtration in order to remove microbes from air or liquids. Here you can see the filter with pores, and this liquid is being filtered through a membrane, filtered through this filter, and you can see the bacteria are stuck on this side of the filter because the pores are fine. The pores are small, and they allow for the water to be filtered. Now, keep in mind with filtering, if it's tiny, tiny viruses the size of the pores, those tiny viruses may get through. So oftentimes, filtering is not necessarily a sterilizing technique. Here you can see that filters can be ultra fine down to 0.02 micrometers. However, even these filters may let the smallest viruses across. So filtering isn't normally a form of true sterilization. Even smaller pore diameters permit true sterilization by removing viruses, even large proteins. You need to have the finest filters in order to achieve sterilization with filtration. Unless you use these ultra fine filters that can even filter out proteins and viruses, the fluid is not considered sterile.
This has the same effect. When you have a lot of sugar, a lot of solute in the environment, it makes it really difficult for microbes to grow. They become plasmolyzed. They become dehydrated. The water leaves the cell. Okay. So now we're going to move from physical control to chemical control, methods of chemical control. And when we're looking at desirable antimicrobial chemicals, what properties are we looking for? We're looking for rapid action, low concentration, solubility in water or alcohol, good shelf life. We don't want our chemical to degrade in just a week or two. We want it to have a good long shelf life. Broad spectrum, that means the ability to kill a broad range of microorganisms. It should penetrate inanimate surfaces to get where the microbes are. It should be resistant to becoming, inactivated. It should be non-corrosive and non-staining. That makes sense. It should have sanitizing and deodorizing properties. It should be affordable and readily available. All of these make it a better chemical to use for control. So these are the desired qualities of a good antimicrobial chemical to use. The high-level chemicals, the high-level germicides, they can actually kill endospores, right? Endospores. Not everything can kill endospores, right? Like even Lysol can't kill endospores. But there are some chemicals that can kill endospores, like glutaraldehyde. You know, hospitals use 2% glutaraldehyde because after 3 hours, remember that? After 3 hours, it does kill endospores. So some chemicals are called high-level germicides. These chemicals can kill endospores. And why would we want to use these sterilants? We could use them on items that can't be put into autoclave, right? We could use them on catheters, heart-lung equipment, implants, things that could get destroyed in the autoclave. Okay. What about intermediate level germicides? These would kill fungal but not bacterial spores. These would be good against some microorganisms, but not all. And we could use these on semi-critical items such as respiratory equipment and thermometers. And then obviously there's low-level germicides. This is probably what you have around your house as in the form of Lysol and other things like that. These eliminate only the vegetative mother cells, vegetative fungal cells, some of the viruses, especially the enveloped viruses, which are more susceptible to degradation. They're used in the clinic on non-critical material, like the electrodes that are placed on your skin, straps or furniture that touches the skin. Okay. And of course, what are some factors that can affect microbicidal activity? Well, what's the nature of the microorganism? Is the microorganism more resistant? Is it more susceptible? Does it have, like for instance, does it have a capsule? Does it have mycolic acids? Is it acid fast? Is it gram-positive? Is it gram-negative? All of these things can affect the susceptibility of the microbe to the chemical, right? The nature of the material being treated, right? Are you spraying a nice, clean, smooth surface? Are you spraying a nice table? Or are you trying to get them out of this cloth material, right? That's going to be a little bit more challenging to get microbes out of this cloth material than it is off a table that you could just spray easily, right? The degree of contamination, that makes sense. How badly is it contaminated? The time of exposure. Remember, we talked about this last time. You can't just spray and then wipe it immediately. Did you allow the proper exposure time for the chemical and the microbes? And then the strength of the germicide. Did you use a high-strength germicide? Or did you use a diluted or watered down or low-strength germicide? And again, we talked about this last time, but what are some targets of these antimicrobial chemicals? The targets are either the proteins of the cell, the nucleic acids of the cell, that would mean the RNA or the DNA, the cell wall, the peptidoglycan cell wall, or the cell membrane. These are the most typical targets of antimicrobial chemicals. So let's talk about some antimicrobial chemicals. Let's start with the halogens. Halogens are elements including fluorine, bromine, chlorine, iodine. All of these can be used because of their antimicrobial properties. They put chlorine, for instance, in swimming pools. That helps to destroy microorganisms that get in the swimming pool. Chlorine is also a central component in bleach, right? Bleach has chlorine in it. Iodine, we all know about iodine. You know, it's very standard to use iodine as an antiseptic. Remember, antiseptic means treating the living tissue, right? Antiseptic. But fluorine and bromine also have antimicrobial properties as well. These are known as the halogens. Chlorine is a very powerful antimicrobial agent. It can even kill endospores. Like I said, chlorine is found in bleach, and bleach is a sterilant. It can sterilize at high enough concentrations. And the way it works, you don't need to memorize this for an exam or anything, but the way it works is it denatures enzymes. It denatures the proteins of those microbes. Okay, this is why you don't want to get bleach on your skin, because it will also start to denature your own proteins and enzymes, right? So you got to wear gloves if you're going to work with concentrated amounts of chlorine or of bleach. And in their swimming pool, obviously, they dilute it, so it's effective against the microbes, but not against you. But it's still, you know, it's pretty toxic stuff. Okay. What about iodine? Remember, iodine, it penetrates the cells, interferes with their metabolic function. It can cause disulfide, or interferes with the disulfide bonds of proteins, so it basically disrupts proteins. And again, you don't need to know each of these different chemicals and what it disrupts, but I'm just mentioning it, you know, just so you know. Okay, but we all know that you can use iodine to help disinfect wounds, things like that, as an antiseptic. Now, if I use iodine on a surface, remember, I'm using it as a disinfectant. If I'm using iodine on my living skin, then I'm using it as an antiseptic, right? You see the difference? If something is used on a surface, it's a disinfectant. If I'm using that substance on the skin, I'm using it as an antiseptic. Exactly right. We don't need to go into all these fine details here about iodine. What about phenol? Let's move on to phenol. Phenolics are these antimicrobial chemicals that have a phenol ring. Phenol was one of the earliest chemicals used to disinfect. Do you remember when I told you about Lister and how he treated his surgical instruments before he did surgery, and he treated the surgical site? He used phenol. Phenol is a potent antimicrobial, but it's pretty toxic, so it's not great. It's pretty toxic. It makes them dangerous to use as antiseptics, you know. So it's kind of like hydrogen peroxide, right? You guys remember hydrogen peroxide was commonly used as an antiseptic, but now dermatologists pretty much agree that you shouldn't use hydrogen peroxide on your skin. It causes a lot of damage, right? Because it's oxidative, right? And phenol's like that. So Lister would have used it on your skin to help prep for the surgery, but nowadays we know that's not worth it. There's better antiseptics to use for surgery. We're not going to put phenol on your skin. But it is a potent antimicrobial. Okay. Again, phenol is a particular chemical, but phenolics are other types of ringed molecules. Phenols are ring structures, molecular ring structures. And these phenolics are known to be antimicrobial. They destroy bacteria, fungi, viruses. And, you know, like I said, they're useful because they're antimicrobial, but they tend to be too toxic to use as an actual antiseptic on your skin. Now, one type of phenolic that we use on the skin is triclosan. Triclosan. And in fact, up until recently, it was very, very common to find triclosan, the phenolic, in soap, hand soap. Anytime you pick up hand soap and it said antimicrobial hand soap, most likely the antimicrobial agent in the hand soap was triclosan. But unfortunately, look what happened here. Widespread use of triclosan led to development of triclosan-resistant microorganisms. So this is terrible. Nowadays, most clinicians would agree that you don't necessarily need antimicrobial hand soap. You just need soap and water, and that gets you pretty clean, unless you're doing something like actually working with microorganisms or something, in which case you might want some antimicrobial hand soap. But just to wash your hands at home, soap and water is fine. You don't need antimicrobial hand soap because that causes a problem, right? Now the microbes get more resistant to those antimicrobials. Is that really worth it? Not really. So, alcohol. You guys saw alcohol in action. Remember when we did our Kirby-Bauer test in the lab? You took the little tweezers, and I said, just put the little tweezers, the forceps, put them in alcohol. And then how long did I say to flame it? Yeah, just for a second. You just want to burn the alcohol off. You know why? Because the alcohol itself kills the microbes. Alcohol itself is antimicrobial. So the only reason we flamed wasn't to sterilize the forceps, it was to burn off the excess alcohol, right? Alcohol has antimicrobial properties, but at high concentrations. The concentration that's usually potent enough to kill microbes is around the 70% alcohol range. That would be 140 proof. So at least 50%, but usually 70%. And if you notice, a lot of these hand sanitizers like this right here says, this is a hand sanitizer, active ingredient, alcohol, 76% volume. 76%. That's basically the only reason this is antiseptic, is because it has 76% alcohol in it. That's it. So instead of buying this expensive thing, you could just mix alcohol and water. But make sure it's alcohol that's meant for use on the skin, because sometimes alcohols, they have like benzene mixed in with them and stuff, so just be careful. Okay. And again, alcohol at high enough concentrations, like this 91% isopropyl alcohol you probably have around your house, people use that as a skin degerming agent, right? As an antiseptic, right? So anytime I get a cut or I get, something, a break in my skin or a splinter, I always treat it afterwards. I wash my hands and I treat it with some alcohol, like a little cotton swab of alcohol. Then place the bandage on top, right? So you want to make sure you wipe off the skin and kill the microbes in that vicinity. And you guys know alcohols are colorless hydrocarbons. You've got the hydroxyl groups on there. Okay, we don't need to know much else about this, but the way alcohols work is they basically attack the membrane of the cell. They attack the proteins of the cell. Okay. And as I just showed you, most alcohol-based hand sanitizers contain about 60-70% ethyl alcohol and are more resistant to evaporation. So they, whatever they mix in there with the alcohol keeps it from evaporating. Because if you've ever worked with like rubbing alcohol or pure alcohol, it evaporates pretty quickly. So you don't want that if it's a hand sanitizer. Okay. Isopropyl alcohol. So ethyl alcohol, this is ethanol. Isopropyl alcohol is more microbial cidal. So it's more deadly to the microbes and less expensive than ethanol. Evaporation occurs more readily, so it's, you have to use it more quickly. All right. And then there are oxidizing agents. Let's move on to oxidizing agents. Remember, oxidizing means to steal electrons, right? Do you remember Leo says Gur? We say Leo says Gur. Loss of electrons means oxidized. Gain of electrons means reduction, reduced, right. So oxidizing agents, they cause things to lose electrons. They steal electrons. Oxidizing agents steal electrons. They cause something to lose electrons, okay? So for example, hydrogen peroxide. Hydrogen peroxide is a good antimicrobial agent because it's oxidative. It steals electrons from things. And when that happens, that causes damage to the cell. That causes damage to the DNA. That causes damage to proteins. It can cause cell death. So again, remember, hydrogen peroxide used to be used as an antiseptic, but nowadays we know that it's not worth it because it's better to use something like alcohol, because hydrogen peroxide, not only does it oxidize the bacteria, but it oxidizes your own cells, and that causes a lot of tissue damage, and it can slow the healing process, right?
When was that discovered? Because I used to always...
Pretty recently, yeah. Pretty recently, but nowadays, I think what they did was they tested, different ways of curing, wounds, curing wounds, and they showed that, with hydrogen peroxide, it takes longer to heal, and so the problem is when you added that hydrogen peroxide, you caused some more damage, and when you cause more damage, it's going to take longer to heal. It's not worth it. So alcohol causes less damage to your own cells. It's better to use as a antimicrobial, antiseptic, I should say. Yeah, as an antiseptic, 3% hydrogen peroxide was used for skin and wound cleansing, mouth washing, bed sore care, but nowadays it's becoming more and more, faux pas to use. The benefits don't seem to outweigh the drawbacks, But in a pinch, if I were in the zombie apocalypse, like Zombieland or something, or Walking Dead, and I had a wound and the only thing around was hydrogen, I would definitely use hydrogen peroxide. But if I have a choice, I wouldn't use hydrogen peroxide. Yeah, at high enough concentrations, we're not talking 3%, but at high enough concentrations, it can be not just bactericidal, virucidal, fungicidal, sporicidal. It can kill everything at high enough concentrations. Okay. Ozone. Ozone is O3, it's a gas. It has similar effects to hydrogen peroxide. It's an oxidizer. And we can use ozone to disinfect the air, water, and in industrial air conditioners and cooling towers. Detergents. Detergents are good because detergents have part polar and part nonpolar ends. That's what detergents have. Part polar, part nonpolar. And that means that they can embed into the membrane of the cell and disrupt the membrane, right? So they often act as surfactants. Remember surfactants? They insert themselves into the membrane, the plasma membrane, and then cause the membrane to disrupt, to open up. There are some limitations to detergents. Ineffective against tuberculosis bacterium, hepatitis virus, Pseudomonas, and endospores. So it's not great. You can't use detergents to sterilize. But detergents are mainly used to sanitize. Do you remember sanitize? Like when you wash the dishes, you use a detergent, right? That's sanitize or degerm. Remember, that's the lowest level of, microbial control is this, just sanitizing. Yes?
So if you had like a towel or something that grew like mold and you washed it with detergent, it would not be...
Not sterile.
So the mold would still be there.
Maybe. Well, there's also agitation. If you put it in your laundry machine, there's agitation, there's hot water, there's the detergent. So most likely the combination of the detergent and the agitation and the constant rinsing, and you're probably going to get most of it off of there. Yeah. But it's not because just the detergent. It's the detergent plus the agitation plus the hot water. Yeah. Good question.
What's the agitation?
Agitation, so unless you don't have a agitator. Newer machines don't have the agitator. They have just like the spin thing, which is, it's not as effective. Surfactant. So remember, surfactants are a type of detergent. Surfactants have a polar end, a non-polar end. They insert themselves in the membrane. Remember this slide? I hesitate to go back this far, but there was a slide here. I just want to show you way back. Here, see these surfactant molecules, remember from an earlier slide 25? These surfactants insert themselves into the membrane of the cell, and then they cause the membrane to disrupt. This is what detergents do. This is how detergents work. They insert themselves in the membrane gets sliced. Quaternary ammonium compounds, we're not going to touch on these much, but they are a type of detergent as well. They're mainly, like soaps, basically. Therefore, they're mildly microbicidal. We did the hand washing activity, right? Yeah, we did the hand washing activity. So with the germicidal soap, you do end up killing more microbes than a non-germicidal soap. But remember, usually for most applications, you could just use regular non-germicidal soap around the house, right? But if you're gonna, cut raw chicken or handle raw chicken or something, then you might want to consider using some germicidal soap to kill the salmonella, for instance. Heavy metals. We kind of touched on this last time too. Heavy metals are by nature antimicrobial because they tend to compete for cofactors in the cell, and that can cause the cell to not function correctly. So heavy metals, what are heavy metals? Mercury, right? Silver, other metals, just metals. For instance, if you took an old school copper penny when it was still made out of copper, right? Let's say you take an agar plate. If you took an agar plate and did a spread plate with E. coli, like you just spread E. coli all over the plate, and then you put a real coin in the, and by real coin I mean one with a heavy metal in it, like a silver coin or a copper coin, and you put that coin in the center of the plate and then incubated and then came back, you would see a zone of inhibition. Isn't that neat? Just like the Kirby-Bauer test, you came back and you saw a little zone of inhibition. If you put a metal, or even a chunk of metal, if you took like a little chunk of silver or a little chunk of gold and you placed it in the center, then the microbes would avoid it. And this is why, it's nice to have water bowls for your pets. It's preferable to have water bowls that are metal for your pet than water bowls that are plastic or something like that, or even glass. And they also say this is why they put the copper plate on the bathroom door. You know, on the bathroom stall, you got the copper plate, because if that's a real copper plate, it's going to have some naturally antimicrobial properties to it. Now, if the previous person just splattered a bunch of stuff on that, that's not going to help you much. But, you know, it is naturally antimicrobial. Right? See, this is what I was talking about. Look at this. This is an actual plate with silver and gold. And look what's around it. Zone of inhibition, right? A zone of inhibition. The microbes want nothing to do with that heavy metal. Okay. Let's move on to aldehydes. Aldehydes are very potent antimicrobials. In fact, you could call them sterilants. That means they're strong enough to sterilize. Especially glutaraldehyde. What do we use glutaraldehyde for? Remember in the hospital, they use a 2% glutaraldehyde solution to spray down inanimate objects for 3 hours, and that's sporicidal. What about formaldehyde? Another type of aldehyde is formaldehyde. What do we use formaldehyde for? Do you know? Preserving things. Yeah, if you want to, you know, if you ever been to a science lab and you see the frog in the jar, you know, it's a preservative. Formaldehyde is a preservative. Glutaraldehyde, formaldehyde, these are types of aldehydes. What they do is they cross-link molecules, they cross-link proteins together, and that can kill microorganisms, right? That can kill microorganisms. And again, like I said, glutaraldehyde, we use this in the clinic. It's broad spectrum. You can use it to sterilize. Isn't that a powerful word? Sterilize? That should tell you something. You know, that means it's the gold standard. You can use it as the gold standard. It's much better than Lysol. Lysol can disinfect. Glutaraldehyde can sterilize. Well, if you leave it on the requisite amount of time, right, 3 hours. You know, you have to leave it on for 3 hours. But if you do, you can sterilize equipment like scopes, kidney dialysis machines, dental instruments, you know, all kinds of things in the clinic.
So it's just like a spray?
Yeah, you spray it. It's a bottle, and usually you just spray it. Or a lot of times they'll have a tub of it, and they'll place the instruments in that tub. Mm-hmm. So again, glutaraldehyde, formaldehyde, these are potent antimicrobials, but formaldehyde tends to be used as embalming fluid or preserving tissue samples, preserving you know, organisms for labs. Skip that. Okay, gaseous sterilants and disinfectants. So you can use gas. You can use gases to sterilize as well. Ethylene oxide is an example. It's a colorless substance that exists as a gas at room temperature. It disrupts DNA and proteins. So it's a strong alkylating agent. Okay. So why would we want to use a gas? So again, remember this is a type of cold sterilization, because if I put something in a gas chamber and then gas it to sterilize it, did I have to heat it up? No. So that's another way to do cold sterilization, right? I don't need to put it in the autoclave. I don't need to heat it up. I can put it in the gas chamber, right, and then expose it to this gas. It didn't have to get hot, right? So it's another way of sterilizing things that are sensitive to heat, right? Medical devices. Let's skip through. Acids and alkalis. Obviously, we know that acids can kill microorganisms. Strong bases, alkalis are strong bases. You know, they can disrupt the cells as well. These cause denaturation. All right. Skipping ahead. Oh, I'm going to skip that one. And that's it. We did it. So we've reached the end of chapter 11. Yeah, pretty straightforward chapter, but an important chapter if you're going to go to the clinic, if you're going to be in the field of nursing, you know, or dental hygiene. You know, you need to know these terms and not mix up these terms. That's one way to be called out as a newbie, is not know the difference between disinfect or sterilize and these terms. Awesome. So let's call it a day. Let's head over to lab. And remember, today's the important, very important